Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore the adaptation of rice for growth in wet environment. At the end of this lesson, you will be able to explain how rice is adapted to grow with its roots submerged in water, limited to the development of parenchyma in roots, ethanol fermentation in roots, and faster growth of stems. Let's get started. Although rice can grow in dry condition, is typically cultivated in paddy fields, which are fields deliberately flooded with water. So the rice has adapted to tolerate growth in water, whereas most competing weeds are unable to thrive in submerged condition. Because most plants cannot grow in deep water, as their root cannot access enough oxygen for aerobic respiration. And the submersion of the leaf prevents photosynthesis from occurring because insufficient of CO2 available for photosynthesis. So what are the features of a rice plant enable them to grow at the submerged condition? First of all, the rice responds to flooding by growing taller. So when submerged, the rice plant grow taller to raise their leaf above the water level. So according to one article, the growth rate is as fast as 10 centimeters per day when the stem is submerged underwater. So when the leaf above the water level, yes, the growth rate becomes slower because photosynthesis can continue by increasing the access to the light and carbon dioxide when the leaf is above the water level. The next features, the stem of the rice plant contain loosely packed cells forming a tissues known as parenchyma. So you can see that in this diagram, we have a lot of these air spaces. These air spaces actually form a continuous uninterrupted channel from the leaf to the root, which allow the diffusion of the oxygen from the leaf to the roots, allowing the roots to carry out aerobic respiration. So these parenchyma tissue provide air spaces that allow oxygen to diffuse from the leaf down to the roots. It's more important to allow O2 to diffuse from the leaf to the root so that the root can carry out aerobic respiration, even though they are submerged under water. Okay, not for CO2 in this case, because we know that the roots will not carry out photosynthesis. So there is no need for CO2 to diffuse from the leaf to the root for the roots to carry out photosynthesis, because the main function of the root is not to carry out photosynthesis. Okay, but yes, parenchyma also serve as another function to allow the CO2 to diffuse from the root to the leaf. Okay, this parenchyma is an uninterrupted channel, okay, for the diffusion. Third, the leaf have the hydrophobic corrugated surface, and some leaf trap air underwater due to the ridge on their surface. If you look at the surface is not flat and smooth. They form the ridge, or what we call the protrusion. So the hydrophobic surface of rice leaf helps repel water, preventing excessive water absorption. And this allowed the leaf to stay above the water level, promoting the gas exchange for photosynthesis. And the trapped air formed the aerial pockets around the submerged leaf, providing a source of oxygen to the plants. Okay, so even though the leaf actually above the water, okay, so because of the ridge, a small air pocket can be formed, allowing the oxygen to diffuse from the leaf into the roots. The next features, same as the yeast, the roots actually produce ethanol dehydrogenase, and this allowed, the roots to convert pyruvates to ethanol in the absence of the sufficient oxygen, allowing the plants to survive in anaerobic respiration. So during anaerobic conditions, the reductions of pyruvates to form ethanol allowed the regenerations of NAD. So the regeneration of NAD allowed the continuity of the glycolysis so that the plant or the rice plant managed to gain 2 ATP molecules per glucose molecule. And the rice can tolerate high level of ethanol. It can be achieved by breaking down the ethanol produced during anaerobic respiration by alcohol dehydrogenase. So this converts ethanol into the less toxic compound, allowing plant to tolerate high ethanol level. So compared to the yeast, as we know, there's accumulation of 8 to 10% of the ethanol to kill the yeast cells. But why this would happen in the rice? Because they have a different type of alcohol dehydrogenase, which break down ethanol produced through anaerobic respiration. So there is an extended learning here to look at the isoform of alcohol dehydrogenase. Many students actually confuse about this alcohol dehydrogenase. Alcohol dehydrogenase is a cluster or a family of this enzyme involved in this formations of alcohol and also breakdowns of the alcohol. So in rice plant, there are two types of isoform. First is known as ADH1, which is an anaerobic isoform, and ADH2, which is known as aerobic isoform. So both of them are alcohol dehydrogenase. However, their main function slightly different. For example, ADH1 mainly involved in the reduction of ethanol to ethanol during fermentation process. So ADH1 actually allows the regenerations of NAD through the reductions of ethanol to form ethanol. This is the main function of ADH1. However, ADH1 also involved in the ethanol breakdown, but primarily this actually is the role of ADH2. So how about ADH2? ADH2, the primary function is to break down this ethanol, okay, to acetaldehydes in anaerobic condition, playing a major role in the ethanol detoxification. However, ADH2 may have a very minor role in the ethanol production. So generally in rice plant, there are two isoforms of ADH. So ADH means alcohol dehydrogenase 1 isoform form ethanol, and at the same time we have ADH2, which break down ethanol. So by doing so, we can actually allow the regenerations of NAD, and as the same time, the concentration of ethanol is not toxic enough to kill the root cell. So with that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any question, feel free to leave them in the comment section. See you in the next lesson. Have a great day.